everyone, welcome to my channel. Today I come with another topic. This is the Southern blot analysis. What is the Southern blot analysis? Southern blot analysis actually process. Here we can identify or recognize the DNA fragments with proper restriction sites. Okay, so you can see here there is a picture. I'll just stick with the board because it's time consuming drawing because we have to I have to complete this video within very little time, so that's why I just stick this picture with the board. Okay, and I explain you. So actually Southern blot analysis is what? This is all DNA that is double-stranded DNA. It contains proper restriction enzyme, and this restriction enzyme remain in a proper site, and that is not similar in every cases. That means that all the DNA fragments, so when the DNA fragments differ, the restriction sites or the location of the restriction enzymes that are also different, and this is the process, Southern blotting process. This is the process by which we can identify the DNA restriction sites. Okay, so this is the process here. First you can see first the DNA fragments are from the DNA strands cut down the DNA fragments, and then DNA fragments are transferred to the electrophoresis process, and then this is called the gel electrophoresis process, and then it is transferred to the membrane. Okay, membrane filter. I explained about the phoresis, electrophoresis and the membrane filter after this explanation. Okay, so and then after that the DNA fragments are transferred exactly as they were to the agarose gel. Okay, and then the DNA fragments complementary to the probe are visible with the restriction sites. Okay, first we learn about what happened here. Okay, the samples of genome DNA first cut and DNA fragment of the different length that depending on the location of the restriction sites. Okay, the length of the DNA that depends on the location of the restriction sites. So fragments are not all the same. Okay, the size of the fragments may be obviously differ. That means each length of fragment is not similar with the other fragment. After cutting down into the fragments, the DNA restrictions fragments are separated by the size using agarose gel electrophoresis. Using agarose gel electrophoresis. This is the process, gel electrophoresis, where we just separate the DNA restriction fragments by this process. After that, DNA is stained with the ethidium bromide. Why it is stained with ethidium bromide? Because without staining or without coloring DNA, we cannot see them under the UV light. Okay, so first cutting them into the fragments, then it is transferred for the gel electrophoresis, and then these fragments are stained with the ethidium bromide. Clear? For seeing them UV light. Third one is the DNA fragments are transferred to the membrane filter, and in brief, the gel is what is the membrane filter is why the membrane filter is needed, or what is the role of the membrane filter. That is in brief say you that here the gel is totally soaked in an alkaline solution to denature DNA. Okay, the DNA, double-stranded DNA here that converts into the single-stranded DNA. Clear? When it is soaked in an alkaline solution. Next, number 4, probe, labeled probe. Actually, labeled probe means all the DNA fragments that remain in the membrane filter, they are not labeled, they are unlabeled. Okay, and here for the binding the DNA, unlabeled DNA with the labeled DNA, it identifies or it recognizes the proper restriction sites or restriction enzymes location. So here what we have to do, one labeled probe is added to the membrane filter. Clear? And what happened after that? this when we just transfer the labeled probe to the membrane filter, it hybridizes to any complementary DNA fragment. Okay, here many DNA fragments are there, and which one is complementary to the labeled probe that hybridize with them. Clear? And the last one is the fragment. From the fragment sizes, a restriction map can be generated to show the relative position of the restriction site. So you can see here the three identified places by which we just identify the unlabeled DNA fragments restriction sites. Here, so Southern blotting process is what? Southern blotting process is in very easy way. We just learn first DNA cut into the fragments with proper restriction sites, so the fragments are not similar all. Okay. Second one, the DNA restriction fragments are transferred to the electrophoresis, gel electrophoresis technique, and here the DNA after the electrophoresis, the DNA is stained with the ethidium bromide. This is one kind of stain for seeing properly in the UV light. And the third one, DNA fragments are transferred to the membrane filter. Here, actually the gel that is consumed by the DNA fragments, these are totally completely vanished from the DNA segment when we just transfer the fragments into the membrane filter. Clear? And actually here the gel is totally consumed by the alkaline solution. Okay? And there is also buffer solution given. I explained it, but first the process you just understand it. Okay, so here the DNA are totally soaked by the alkaline solution, and it denatures into the single strands. Okay, the double stranded DNA that transferred to the single strand, and then one in this membrane filter, when the DNA fragments remain in the membrane filter, same one labeled probe that totally identified or recognized with proper restriction enzymes, this is transferred to the membrane filter. And what happened there? The already present the DNA fragments within the membrane filter, which one is the complementary this labeled probe that hybridize with this probe. Okay. Now we can easily recognize the map or sizes of restriction map. Okay, of the DNA fragments. So this is the process of the Southern blot method. Okay, and here I just explain you about the electrophoresis and the membrane filter. What happens or what is the proper method of the membrane filter or the electrophoresis? I just explain here now. Okay, now we learn what happened in the gel electrophoresis. Okay, first. One second. Where we just transfer the DNA fragments into the gel, or we just run through the gel for the electrophoresis. What happens? A smear of fragments is seen all over the gel. Here, this is the gel. You can see this is the gel. This is the gel, and gel that contains all over there the smear is formed, and this type of this is gel and there is a smear. This is a smear of the DNA fragments, all the DNA fragments that ranging from large to small. Okay, the large lengthy fragments remain in the upper side, and the small fragments of DNA that remain in the lower portion. In this way, we can see a smear of the fragments in the gel. Okay, so this is the process of the electrophoresis, gel electrophoresis, where we get the gel with the DNA fragments with different restriction sites. Clear? And now we understand the membrane filter. What happened in the membrane filter? Because this we get this gel with the different types of fragments, and in such a manner that we cannot recognize them properly. Okay, so this gel is not necessary for the recognizing the fragments. So what we need to, first we need to separate the fragments, and as we just separate the fragments, then when we put the probe, that is the labeled probe, when we just put the labeled probe, then particular DNA that is complementary to the labeled probe, they hybridize with each other, and then what kind of we get the proper membrane filter, like this here, this type of, okay, like this. So what happened in the membrane filter? Actually, here the gel is totally neutralized and placed on a piece of blotting paper. This is, you can see this picture here. This is the tray, and it contains the buffer solution. Okay, this contains the buffer solution. Just upper this blotting paper remain. Okay, this is the tray with the buffer solution, and this is the blotting paper. This is the gel. Upper side of the blotting paper we put the gel, and after that the membrane filter is there. Okay, this is the membrane filter. Next, on the upper side of the membrane filter, this one is the paper towels. Okay, paper towels, and this is the weight. What happened here? When we just put the gel for the neutralization, the blotting paper on a glass plate. Okay, the blotting paper, there is a glass plate must be there, and the blotting paper is there. Okay, and in a container, and the piece of the membrane filter is laid on so that it covers the gel. Just covering the gel, you see that this is gel, and covering this gel, the membrane filter. This is the membrane filter. You can see this is membrane filter. This one is just put or kept over the gel, as in such way the total gel, the total imprints of the gel DNA fragments that present in the gel, that imprints on the membrane filter. Clear? So here the sheets of blotting paper and the weight are stacked just in such a way, and after some time the DNA fragments that remain in the gel, this transfer to the membrane filter. Okay, this transfer to the membrane filter, and after that, when the fragments are totally transferred to the membrane filter, then we put the another probe that is already labeled, and here the labeled probe just hybridize with the proper its complementary fragment. Okay, and in this way we can get a proper map of DNA fragments with proper restriction, enzymes and length. So this is the southern blot method. Same process is used in the northern blot method. If you can understand in this manner how the gel electrophoresis or the membrane filter is used for the transferring of the DNA fragments, then you can easily understand the northern blotting method.